So, welcome everybody to the afternoon or evening session of the ICTS program. Today's speaker is Alex Mogilner, a biophysicist, theoretical biophysicist who is famous to get the numbers right and to collaborate very closely together with experimentalists. And today he will talk about a topic which is relevant for many fields, about the speed and accuracy of mitotic spindle assembly. I mean, something like spindle assembly is also relevant in other areas of cell biophysics, like migration, polarization, polarization during mTOC relocation for immune cells and other things. So I'm very much looking forward to his talk. Please, the screen is yours.
Thank you for kind introduction. Let me share the screen if I can. Yeah. So, all right. So I'll talk today about mitotic spindle for a couple of reasons. So one is my former couple of postdocs, Raja Paul and Ambarish Kunwar, are in India, and I saw them in the audience, and they participated at certain stages of this work in the past. And another reason is that this conference is on statistical physics and biology. And so the example of mitotic spindle is, it's not exactly statistical physics because we don't have the numbers in the spindle as you'll see. So we don't have thermodynamic limits, we don't have 10 to the 23 molecules. We have tens of molecules. But you will see that there are certain features of statistical physics that are present in this exciting problem. And by the way, thank you very much for being here, for listening to me. I wish it was in real time and in real space. I love India. I've been there many times in the past. So hopefully in the future, I can speak to you directly. So these are the people who were involved in the project I'm going to talk about. So Chris Miles is my postdoc at Courant Institute here in New York, who did all the modeling, and he actually did very ingenious analysis of the data. So most of what I'll be talking about is actually not mathematics and physics, but data analysis, which is actually becoming more important these days than mathematics and physics. And we collaborated with a wonderful experimental lab of Alexei Kodjakov, who is actually in the State Department of Health here not far from New York. And actual experimental work was done by Ferran Arenda, Alexei's postdoc. And also we'll mention briefly one of the results that that is part of the project I'm doing with this lab at University of Massachusetts. So I'll be talking about mitotic spindle as advertised, and I wanted to show in the beginning this part of the mural, which can be seen in many museums all over the world. This mural was made by Diego Rivera, actually exactly 100 years ago. It's called Man at the Crossroads. There are many interesting social themes in this mural, but what I wanted to draw your attention to is this depiction of the spindle right at the center of the mural. This drawing was done by Diego Rivera after famous drawing of Ramon y Cajal, a Spanish scientist who did this wonderful artistic drawing of many cell organelles more than 100 years ago. And here in the corner of the slide, you see actual snapshot of the spindle, very familiar to pretty much anybody who do any kind of science. So it looks like American football. This characteristic spindle shape, it has two poles with two centrosomes in the poles. It has the chromosomes in the middle and microtubules connecting centrosomes to the chromosomes. But I'm jumping ahead here. So there are many super interesting and exciting problems associated with the spindle. Like some of them, for example, how does the spindle gets its shape, right? Another, how do chromosomes get aligned so precisely at the equator of the spindle in metaphase, in the middle of the spindle life? How do these chromosomes get separated to the opposite spindle poles? And so on and on and on. So today I will address one of those questions, which is absolutely fundamental. It's actually how does the spindle self-assemble at the stage called Prometaphase. And the question is actually how does it assemble so rapidly and accurately? You will see at the next slide what the nature of the problem is. So there are roughly speaking two big paradigms, two big concepts of how spindle assembles. So one concept is that what is crucial for spindle assembly is chromosomes with kinetochores, the special organelles at the centromere of the sister chromatids, which are for physical purposes are like molecular glue for the microtubules, dynamic fibers that grow from the centrosomes. So we have two mitotic asters. Centrosomes serve as anchors for microtubules, and we have a few tens of the chromosomes, and then somehow a connection between the plus ends of the microtubules and kinetochores. Those connections are made, and we have the spindle. So this is the so-called centrosome-dependent spindle assembly. So there's an alternative model, which is acentrosomal spindle assembly. This model says that we have, again, chromosomes, we have microtubules, but the centrosomes are either not necessary or kind of secondary importance. And then somehow chromosomes and these microtubules self-organize into the structure, right? And there's, of course, possibility that we have some kind of hybrid assembly, that microtubules and chromosomes self-organized, but also there's this microtubule asters get connected to the chromosomes, and we get the spindle. So let me start at the beginning. The fundamental discovery that pushed forward these ideas of how spindle got assembled was made about 35 years ago by Mitchison and Kirshner, when they observed that in the cell, microtubules undergo what is called dynamic instability. So they grow and shrink rapidly and repeatedly. And immediately after this discovery, Mitchison and Kirshner started to wonder, what is this behavior for? And they came up with a beautiful hypothesis called search and capture. And this hypothesis says that the spindle assembly is as simple as that. In the beginning, we have this sister chromatids with kinetochores and hundreds of microtubules doing this dynamic instability in space, absolutely blindly and in an unbiased way. And then once in a while, a microtubule is growing in the right direction, meaning that it's growing toward the kinetic core. And then when sometimes this growth ends in microtubules bumping into the kinetic cores and connecting to it, right? And that's the event called capture. And then you have one of the kinetic cores connected. Then another kinetic core, sister kinetic core, gets connected the same way to the opposite pole, and that's what we need. That's what the correct capture called amphitelic capture. But then you can imagine that if you are doing this process repeatedly and randomly, what you get is a mixture of correct attachments like this and erroneous attachments like this syntelic attachment, like this merotelic attachment, right? And so the question now is probably becoming clear. We have hundreds of these microtubules. We have tens of the chromosomes. And obviously, if you want to use this process to capture all the chromosomes, right, you have to wait, because the first lucky chromosomes probably get captured rapidly, but then there are some unlucky chromosomes that just by chance will be found later, and so this process is probably going to be lengthy. And then it's kind of easy to imagine that there will be a lot of errors like that. So obviously, all these questions are quantitative. So this is one of the wonderful problems in cell biology, which definitely requires physics and mathematics to understand. And the first beautiful calculation was done by Holley and Weibler in 1994. There was actually also a paper by Terrell Hill in the 80s, which was only kind of vaguely about the search and capture. So what they did, they found what are the optimal dynamic instability parameters to get fastest search and capture. But they did it in a very physical way, kind of with the back of the envelope estimates, with one microtubule searching for one target. And so at some point with my former student Roy Wallman, we decided to just do direct Monte Carlo simulation. So we placed chromosome in a random position and looked actually many chromosomes in random positions and looked how fast hundreds of microtubule can capture it. And after that, this search and capture model undergone quite a number of developments and iterations, and we gradually were making it more and more realistic. And that's largely the work of Raja Paul, who is now in Calcutta. And his beautiful work ended up in a couple of very interesting predictions. So one of these predictions that because of this randomness of the search and capture process, and because of this problem that we have many chromosomes, and the last chromosomes to be captured, you have to wait for them for a long time. Then the first chromosomes would be captured relatively rapidly, but then there's this long tail in this distribution of chromosomes to be captured. So you have, so the cell has to wait for the last kinetochore to get captured before it can go to the anaphase. So you have to wait for a long time. Even more important predictions from this models was that there'll be a lot of errors in this process. In fact, the model predicts that majority of the attachments will be merotelic. And the problem here is that, well, obviously, the first attachments will be monotelic, then there'll be many syntelic and amphitelic, but you wait long enough and you will get some of the merotelic attachments. You simply start collecting the wrong attachments. Of course, the cell doesn't do it in this completely random and damp way. There's many tricks the cell does to prevent and correct errors and to accelerate the search. And so all these papers I listed here were our attempts to refine the search and capture model and explain how this process could be accelerated and be done more accurately. So, for example, what the cell does, the kinetocores actually sense the tension across them. And the tension results from molecular motors on the kinetocores, mostly dyneins, trying to transport the kinetocore to the minus end of the microtubule at the centrosome. And so they basically pull the sister kinetocores in the opposite way, stretching them, and the cell senses the stretch and solidifies amphitelic attachment. While without stretch, this attachment is unstable, microtubules just detach. And there are many other tricks that the cell does. So the latest iteration of the search and capture model is like that. So we embarked recently on absolutely humongous simulation on a supercomputer in which we model all the microtubules which are now elastic and can bend. And sister kinetocores are connected by effective spring. Then we have proper geometry of the kinetocore in space. And we have all the actual cross-linkers and molecular motors on the each kinetocore interacting with the microtubules, and we have Aurora-B molecules, kinase, which participates in this error correction process and many other things. And we also have chromosome arms, right? And so I'll show you briefly the simulation, which takes almost a day to reproduce about 30 minutes of biological times. And you can see all these tiny kinases and molecular motors near the kinetocores. You can see all this, the whole process. And this is only one chromosome shown, but we cannot simulate tens, but we can simulate up to tens. And so the result of this simulation shows that we can get close to what biologists observe, right? Which is that the assembly of the spindle takes basically like 10-20 minutes, and the amount of errors is really minuscule. But it still cannot quite get there with the optimal parameters. There's still a number of errors is in tens of percent. We have 20-30% errors, and we do have to wait significantly longer than 20 minutes. So this search and capture model is more and more in doubt. And so I will show you by the end of the talk that that some parts of the search and capture models are surviving, but there's something else which is very important, which is happening in the spindle. So what we were missing this whole time is information on where centrosomes and chromosomes and kinetocores are all this time that the spindle is assembling. So for decades, it was impossible to get this data out of the cell. And finally, literally decades of heroic efforts of cell biologists resulted in this result, which is we now can track centrosomes and kinetochores in space, in three-dimensional space and in time, with very high spatial and temporal resolution. So the spatial resolution is sub-micron, and temporal resolution is we have positions every 5 seconds. So what you see here in this movie is a view of the cell from 3 angles. So this one is from above, and those 2 are from the side, right? And what you see is that centrosomes that are initially at the south and north pole of the cell, and chromosomes are all bunched around them. So then centrosomes separate from each other and move along the surface of the cell to the opposite points on the cell equator. And you see that there's something important happening with the chromosomes. One thing you could see that the distance between sister kinetochores is get stretched, and the chromosomes are all aligned along the spindle. But just by eye, it's kind of hard to see everything that happens. So these days, of course, you can automatically track many quantitative characteristics of the spindle. So 2 important characteristics of the spindle are as follows. One is the interkinetochore distance, and another is the so-called C-tilt angle. It's the angle between the vector of the centromere of the chromosome, the vector between 2 sister kinetochores, and between the spindle axis, the segment between the poles, right? And of course, we can measure pole distance, and we can measure many other things. So if you look at those measurements, we did those for tens of cells. So we now have good data for tens of cells and more than 1,000 chromosomes altogether. And then we can look, of course, cell by cell. And so what is key to for understanding this data is you, at the same time, look for the whole ensemble, and then you look at individual cells. Excuse me for 1 second, I'll be back in a second. All right. So, this is the characteristic time series for pole distance. So you saw that centrosomes are starting close to each other, and then they segregate. And these are the curves for 2 random cells from this ensemble we have, cell 1 and cell 2. And what you see is that those 2 cells, they segregate the poles to slightly different distances, and the segregation is finished at different times. This cell is delayed. Now, when we observe the interkinetochore stretch and the C-tilt angle, what we see is very simple. So the kinetochores gradually get stretched. So that makes sense. Sister kinetochores are captured from opposite poles, and they are pulled in opposing distances. There's effective elastic linker between them, so there's this stretch. And alignment of the chromosome with the spindle axis is also relatively clear, right? If you pull from the poles, you clearly will align kinetochore vector with the pole vector. And so we then quantitatively define bi-orientation of the chromosome as coincidence of two events. So one is that the kinetic distance becoming larger than some threshold, which is about 0.9 micron. And the CTL angle is becoming lower than something like 35 degrees. Right? So we can, of course, get the same data for each individual cell, the data becoming a little bit more noisy. And so we have such data for many, many cells. Now, here's the first important piece of data which throws search and capture model in doubt. This is the histogram, two histograms for two random cells of the times that at which bi-orientation takes place. And what is very important is how we measure this time. So we measure this time not in, we measure this time in the experiment in seconds, of course, but then we rescale the time, normalize it in such a way that one time unit is the time when the spindle elongation ends. So for each individual cell, so for example, for this cell, this is the time one. For this cell, this is time one, right? So this time is different for different cell, the physical time. But then after normalization, what you see is that the vast majority of the chromosomes get bi-oriented at certain fraction, like at 0.7 of the time unit. So there's very little difference between these two cells. And when we look at the ensemble, same thing, there's very little difference between this time. And also, if you look at the histograms, they are relatively similar to a Gaussian, right? Which is a very important indication that this process of bi-orientation, it is not random, right? So search and capture model says that this process is random, hence this really long tail in the probability distribution of times of bi-orientation. But the experiment says it's not random. It's pretty much deterministic with some noise, of course. And this deterministic event somehow is connected with the distance between the spindle poles, meaning it's something connected to the development of the spindle, of the elastic cage of microtubules between the two centrosomes. So here are the three-dimensional, well, this is two-dimensional view from above of the trajectories of the two centrosomes, which separate from each other and they do some little complicated dance, and of all the tens of the chromosomes in the cell. Right? So it's a very beautiful picture. It's kind of looks like an art. And what you can see from it is that at the initial stage, chromosomes move centripetally toward the center of the developing spindle. And then they start moving along some barrel-shaped surface, right? So first they move centripetally toward the spindle, and then they start moving along curved surface. So to make sense of what's happening, first it's good to change the coordinate system and to observe what's happening not in the immobile log coordinate space, but in the coordinate system which is aligned with the spindle. So basically the vector between two centrosomes is becoming z-axis, right? And you use cylindrical coordinates, so then the radial distance is from this axis, and you have this angle around the axis. And so in this coordinate system, you can kind of see better that poles separate from each other, chromosomes first move centripetally in and then start moving along this surface. Now, this is kind of the biggest experimental discovery. We were stunned when we see what's going on. Is the following. Remember that we can track the bi-orientation for each chromosome. We can detect at which time point chromosome become bi-oriented, correctly attached. So what we observed is that vast majority of the chromosomes get bi-oriented right when they arrived at this barrel-shaped surface associated with two spindle poles. So these are trajectories of two peripheral chromosomes that were far away at the periphery of the cell in the beginning, right? And so this chromosome moves, and this trajectory stops when a computer detected that this chromosome got bi-oriented. And see, it got bi-oriented on this hand-drawn surface. That's another example of another chromosome doing the same. Now, some chromosomes, of course, are very lucky from the beginning. They are already close to the future spindle. They actually don't move anywhere. They just sit there and wait. See, they sit there and tremble and then get bi-oriented, but they get bi-oriented also when there's when spindle is at a certain stage. So see, what is remarkable is that chromosomes from different parts of the cell got bi-oriented at the same time, roughly, right? And not only do they get bi-oriented at the same time, they arrive at this magic surface at the same time. So this is certainly against the search and capture model. Search and capture model would say that those chromosomes get captured first, those chromosomes get captured last. No. So this shows all the close to 1,000 chromosome in different cells for normalized position of the spindle poles, and see the majority of the chromosomes get bi-oriented when they arrive at this certain surface of a barrel. So at this point, we started theorizing. Now, I don't have that much time, and so I will describe to you this only a sketch of a model. So the key to the model is that we know from a previous study, which was done a couple of years ago, that there's many short microtubules. So in the spindle, there are not only this long centrosomal microtubules that extend from the centrosome as an aster, but also lots of short microtubules, and the short microtubules are in the vicinity of each kinetic core. And there are roughly 20, 30, 40 short microtubules near each kinetic core. So we don't know almost anything about the origin of those microtubules, but they don't seem to be growing from the kinetic core like many studies suggest. They're just around. So important thing is that in the healthy wild-type cell, many of those microtubules are eventually arranged in such a way that the plus ends are on the kinetic core and minus ends are sticking out. And this is very likely the work of the motor called CENP-E. It's one of the kinetins, plus end directed motor. So obviously, what the kinesin motor does, it tries to attach to the microtubule and transport it in this way, so that eventually plus end is at CENP-E. And there was an important study in the past showing that when the plus end is reached, the motor doesn't just eject microtubule, but rather grabs the plus end and holds it. Okay, and so that's how this is happening. And there were a hypothesis before that. Then what happens is that dynein motors, they move to the minus ends of the short microtubules and stick to the minus ends, and then those dyneins start interacting with centrosomal microtubules and transport short microtubules to the minus end. As a result, the whole kinetochore and chromosome with it is pulled to the spindle pole. Now, there's also dynein on the kinetochores. And this dynein also can transport the kinetochore to the minus end of centrosomal microtubule. But this dynein also, of course, can mess up a little bit this process, proper alignment of the microtubules, because dynein will try to move the microtubule so that its minus end is near the kinetochore. So this will be broken a little bit. Now, knowing all that, what we propose is the following: that initially, when the spindle is very nascent and chromosomes are still far away from the spindle, then long centrosomal microtubules from both poles interact with the short microtubules on each kinetochore, right? And as the chromosome approaches the spindle, and as the spindle grows toward the approaching chromosome, then something like this happens, right? Now, this is not good, because this connection is pretty good, right? It's pulling this kinetochore to this pole, this one pulling to this pole, but there's also this wrong connection. So what we need is to break the wrong connections. So what we suggest that somehow these wrong connections get broken, only proper connections survive, and then eventually this microtubule will grab that kinetochore, this microtubule, that kinetochore, and we get this proper bi-orientation. Now, to explain this miraculous breaking of the wrong connection, we propose this tug-of-war model. So those kind of models were invented by Lipovsky and his collaborators, and then Steve Gross and Ambarish Kunwar and myself, we worked on those models as well. And so the classical tug-of-war models is when two opposing motors try to move cargo in opposite directions. So here we have somewhat more complex and more tricky situation. So we have two sister kinetochores connected by effective elastic spring, right? CENP-E holds short microtubules with motors connecting to them on the kinetochores. And so then dynein motors on the end of this of those microtubules, and microtubules together with those connected motors we model again as effective elastic spring, are trying to move to the minus ends of the microtubules to which they're attached. So here the tug-of-war is between the same motors, between dyneins. But those motors move on opposing microtubules, and so pull two respective cargos, and cargos are attached elastically in the opposite way. Now, those are equations of the model that we simulate numerically. What I was going to say, right, so the movement is governed by... Is there any...
Das muss sein, ja.
alles da, aber da steht ja schon DFG und so.
Ja, das ist doch okay. SFB gehört zu DFG, ist alles DFG.
Das muss alles da noch dazu.
Ja.
So is there a question or something with the sound? No? Okay, I'll continue then. The movement is governed by force velocity curves, and there are, of course, force balances for all these elastic springs connected together. Now, the key part of this model is the rate of detachment of these motors from the centrosomal microtubules. So this rate of detachment first is force dependent, and this is the standard feature of this force balance tug-of-war models. So if the force is large and the system is stretched, then the detachment is faster. But more importantly, microtubules pulling in the wrong direction, meaning that they are pulling kinetochore not perpendicular to its surface, but at a weird angle, they get detached. And what we're thinking about is that the microtubules are broken from CENP-E motors. There were many studies showing that the detachment depends on the angle at which you pull microtubule from respective motor. So when we simulate this model, and so in this simulation, those segments are dyneins. These are short microtubules. Circles mean that this microtubule is short microtubule is searching for the long microtubule. This is interkinetochore spring, so in this simulation, this is minus and this is plus and minus and plus and. So this is anti-parallel bundle of microtubules, right? And it beautifully shows that initially there are many right and wrong attachments, but eventually only right attachments survive. So those attachments that pull each sister kinetochore only in the proper direction, right? And you also see the proper connection kind of starting to form. Now, we can also simulate, of course, many different arrangements. So in this case, all these microtubules are coming from the pole which is here. Those microtubules coming from the pole which is here. So there's this two system of microtubules intersecting in a perpendicular way. And in this case, biorientation does not quite happen. So the key here is that chromosome has to interact with anti-parallel bundle of microtubules. Now, model is super simple, right? But it gives a number of very simple and specific predictions. Prediction 1: chromosomes bi-orient when they hit the spindle, right? Why? Because before they arrive at the spindle, chromosomes see this arrays of microtubules that are not anti-parallel. Anti-parallel microtubules are only on the spindle itself. Second, after hitting the spindle, it takes tens of seconds to achieve biorientation. Why is that? Because in this tug-of-war model, we know roughly all the parameters from the experiment. It takes like roughly 1 second for each motor to attach and detach. There are tens of the microtubules, probably the order of 10 of tens of short microtubules, order of 10 long microtubules each kinetochore sees. And so the simulation predicts that it takes about a few tens of seconds. Number 3, what if we inhibit dynein on the kinetochores? What is important here is not dynein in the whole spindle. Dynein is so important in the spindle life that if you inhibit the whole dynein, mitosis goes, the whole mitosis goes wrong. But you can inhibit only dynein on the kinetochores, this dynein. So inhibit this dynein, and you leave this dynein working. This is done by knocking down some protein which connects dynein to the kinetochore. So if you inhibit dynein on the kinetochore, what we predict is that chromosomes get to the spindle slower, but after hitting the spindle, bi-orient faster. Why is that? Well, because probably part of the process getting to the spindle is being transported on the long microtubule by dynein. So if you inhibit this dynein, you will have harder time arriving to the spindle. You can just sit where you are and wait for the spindle to grow to you, which we actually see happening. But after you get to the spindle, see now this dynein, it's not there. It doesn't compete with CENP-E anymore, doesn't engage in tug-of-war with CENP-E. And CENP-E has easier time properly aligning short microtubules. So there are more proper short microtubules, more interactions with long centrosomal microtubules, and faster bi-orientation. And finally, if you inhibit CENP-E on the kinetochores, then chromosomes get to the spindle as fast as in control, right? Because CENP-E has no role in moving chromosomes to the spindle. But after hitting the spindle, the bi-orientation will be slower because then the short microtubules get somewhat disorganized. So remarkably, all these 4 predictions actually turn out to be true. So first, chromosome bi-orient upon hitting the spindle. So our collaborators imaged the spindle in time as the spindle is assembling, and quantified the shape of the spindle with those arcs. Right. So here the colors is green is microtubules, and red is PRC1, the protein which cross-links antiparallel microtubules. And so then, if we juxtapose the outline of the spindle at different time intervals, so from early to later stages of Prometaphase, and coordinates of the chromosomes which undergo bi-orientation in this time interval, you see that those bi-orienting chromosomes are crowded around the spindle surface. Okay, so what I showed you earlier in the talk, that chromosomes arrive at a barrel-shaped surface and get bi-oriented. So this surface is the spindle. Okay, so this is the main discovery, that the deterministic process of bi-orientation, chromosomes somehow get to the spindle, and upon hitting it, within minutes, get bi-oriented. So second prediction, that after hitting the spindle, it takes tens of seconds to achieve bi-orientation. This is also turning out to be true. So we have close to 10 micrographs like that. So we observe in real time in live cell the microtubule bundles of the spindle. So this is between 3 and 4 minutes. And under higher magnification, you can see these 2 sister kinetochores, which at 3 minutes after beginning of the Prometaphase are arriving near the boundary of the spindle. This is big bundle of the microtubule bundle of the spindle. And you see within a minute, this kinetochore pairs turns, aligning with the spindle, and stretches. Okay. When I first saw a sequence of images like that, my mind was blown. It's quite remarkable that the model predicts this tens of seconds, and this is actually happening within a minute or so. I'll later show you the statistics because like these cherry-picked images, of course, can be a little bit deceiving. So that's the statistics. So the prediction is this: if dynein on kinetochore is inhibited, chromosomes get to the spindle slower, but after hitting the spindle, bi-orient faster. So this is the histogram of time of bi-orientation. Again, see this beautiful Gaussian shape in control. This is bi-orientation and dynein inhibited spindles see longer time before bi-orientation. Now this is time of hitting the spindle, and this is a normalized time where 1 is time of elongation of the spindle. So time to hit the spindle is shorter in control cells than in dynein inhibited cells. And then time from hitting the spindle edge to bi-orientation is, on the other hand, longer in control cells, exactly like the model predicts. If you look at the CENP-E inhibited spindle, the total time of bi-orientation is very similar in control and the inhibited cell, but the time between hitting the spindle and bi-orientation is longer in CENP-E inhibited cells, again as the model predicts. Okay, so that's pretty much my story. Here's the general philosophical lesson. So the previous thinking was that we have this search and capture model that the central players are chromosomes and 2 microtubule asters, and then the asters connect to the chromosomes. So the twist on this story, actually radically changing the story, I would say, is that this previous model ignored what is actually the spindle, the anti-parallel microtubule bundles, which are assembling at the same time with capture of the chromosomes. And this central spindle, it plays crucial role in attaching chromosomes to the spindle, as we now understand, right? So this is the picture. So somehow the spindle is getting assembled at the same time with getting the chromosomes to the spindle. And then when each chromosome gets to the spindle, it doesn't make end-on attachment right away. First, those short microtubules around the kinetochores get sorted. Their polarity gets sorted by sister kinetochores and long centrosomal microtubules in a process of a tug-of-war. And this kind of primes the kinetochore for eventual correct amphitelic end-on attachment. So that's the story we're telling. Now, of course, many puzzles remain. This is just the beginning of a long journey which we have to walk. Before I tell you actually those puzzles, let me also mention that what we are saying now is that philosophically speaking, we have the hybrid model of spindle assembly. It's neither purely centrosome-centric nor acentrosomal. There's elements of centrosomal driven assembly and acentrosomal assembly. It's actually a union of those two mechanisms. So many puzzles. I'll just mention a couple. So one is search and capture actually still survives because somehow these chromosomes with short microtubules near the kinetochores first have to get to the spindle, right? And they probably have to be captured by this long centrosomal microtubules and brought to the spindle. how does this happen? Well, it could be pure search and capture, meaning that this long microtubule captures individual kinetochores or short microtubules. We are now starting to investigate this topic. It turns out to be much more interesting than we thought, for the reason that there's actually turns out to be relatively few of the astral microtubules like that from the very beginning. Certainly not enough to bring synchronously tens of the chromosomes at the same time to the surface of the drawing spindle. So there are some collective interactions between the chromosomes that are going on, and we actually have a little project with Roger Paul right now looking at this synergy between chromosomes searching themselves and two central searches of the central cells. Another even bigger puzzle is how do you convert this initial messy structure of the short microtubules connecting to the long microtubules, short microtubules connecting to the kinetochores into the proper end-on attachment when you have long microtubules bundles with plus ends connecting the kinetochores. Right? So it seems to be very delicate and elegant and highly regulated process, and that's another thing we are investigating right now. So I wanted to end with again drawing your attention that many themes of statistical physics are here. Basically, how do we move from completely random stochastic events into very predictable deterministic result? And the beauty of the spindle is that spindle does it without thermodynamically large numbers, with just tens and hundreds of molecular pairs. It does this absolutely beautiful, correct self-assembly very faithfully. In tens of thousands of cells, there's 1-2 errors. So this speed and simultaneous accuracy at these small numbers of stochastic pairs is absolutely remarkable. And so the spindle is, of course, not the only organelle in the cell that has this feature of very rapid and accurate molecular process. And hopefully, this design principles of combining the speed and accuracy at small number of molecular pairs are starting to emerge. And this is a great future challenges for us as physicists, mathematicians to contribute to the experimental discovery process in trying to understand those principles. Okay, thank you. And I am ready for questions if there are any. I wonder, I probably should stop sharing, right? Let me stop sharing so that I can see the screen. And I see the raised hand. Please, let me actually.
Thank you very much for this wonderful. Via Krishnamurti, he raised his hand. Please go ahead.
Thanks a lot for the very nice talk. So I was wondering, maybe you said this, what sets the size of the spindle? And in particular, I'm thinking if such models can explain the scaling of the spindle that one observes in different size cells.
Yeah, this is a great question, but the answer could take another hour. A lot about it is known, and there's ongoing research on it. So shortly speaking, there are interspindle forces inside the spindle, molecular motors cross-linking these anti-parallel microtubules. And their tug-of-war or force balances decide what the length is. Another factor is astral microtubules grow to the edge of the cell, to the cell cortex, interact with dynein there, and position the poles. So in different types of cells, there are different balances of those two things. For example, just a month ago, there was absolutely beautiful paper from the group of Needleman and Shelley published in eLife, showing that in C. elegans, these forces inside the spindle are absolutely irrelevant, that it's really pulling of the poles to the edges of the cell, which decides what spindle size is. Our old work in Drosophila spindles says that, in fact, no, those dynein governed forces are not super important, but forces inside the spindle are important. So short answer is basically force balance between many opposing motors, but specifics depend on the cell type.
Thanks.
And I see that, and I see that in the chat, right? There's a question. After chromosome hitting the spindle and orientation, how likely is the formation of merotelic attachment and correct subsequent correction? Excellent question. So we have some data on it. And the answer is, so in the wild type cells we have, and mind you, these wild type cells are in a very bad circumstances. So they, of course, are slightly photodamaged, they are flattened. Even in those perturbed cells, the probability of merotelic attachment is extremely low. We have statistics, it's about 1 merotelic attachment per 10 cells. So, so basically 1 out of a few hundred chromosomes get merotelic attachment, which invariably get corrected later in the during metaphase. If we inhibit CENP-E, then there are way more merotelic attachments, and some of them don't get corrected. These cells get damaged in mitosis. If we inhibit dynein, again, almost no merotelic attachment. So theoretically, we have no good understanding of that, but that's the answer to this question. Okay, so another question from Apurba. What are the nature of the short stray microtubules? Yeah, as I said, we don't know almost anything about them. They don't seem to be growing from the kinetochore. So our favorite hypothesis is that there's this RAN GTP protein, which is, there's this cloud of this protein diffusing around the chromosomes, mostly actually centromeres. And so those proteins activate nucleators of the short microtubules. So we think that short microtubules just appear in the cytoplasm from, of course, not by themselves, from gamma Turk, which is floating in the cytoplasm activated by RAN. Are they static and dynamic? Again, we don't know. They're very likely dynamic, but probably not as dynamic as long microtubules. That's another big mystery for the future. Debashish is asking, is there any similarity between models and Eva-Tolich models? That's also a great question. Yes, there's, I would say, well, it's very superficial similarity, but certainly we think that the short microtubules are becoming the seeds for these bridges that interconnect long microtubules, and that's a big discovery of Eva-Tolich of the last few years. So very likely it's kind of, that's where our stories intersect. Yeah.
Okay, okay, thank you. I
have one question concerning the short empties. Do you actually see them in under a microscope?
Yes, I didn't show those data because it's published, but yeah, there's both the light and the electron micrographs showing these microtubules. There's some image data for the polarity. Yeah, so it's not fantasy. Yeah.
And you can extract statistics. I mean, it would be interesting. I mean, to see how they orient as a function of time.
Yeah, it's all in this paper published in 2018. So there's some data that they align, some data that they align perpendicularly to the chromosome surface. Yeah, there's some statistics.
Thank you.
Please, I see two raised hands. Please ask in any order.
Yes, thank you. I had a question about this K ingredient that dynane detaches more easily when it's pulling in the wrong direction. You mentioned that we know that this is possible. What kind of experimental facts do we have? Because I know a lot of data for the detachment on the head side, but from the tail side, because it's detachment from the kinetochore, right?
Yeah. Yeah, so I was kind of very optimistic and superficial, saying that I think I know the same data you know, and we just extrapolate. So we're basically saying that it's possible that you pull at the wrong angle, you detach without specifying is it detaching from the head of the motor, the tail of the motor, which motor. So this part of the model is totally the weakest. You can really consider it as a unsupported assumption.
But you would say that the data we have for detachment may be actually tail detachment. We don't know.
Yeah, we don't seriously know. Yeah.
Okay, interesting. Thank you.
Sure. It was another
Okay, currently I do not see any more questions.
I have a question. Heiko.
Yes.
Yeah. So this is a very general question for for all work on this search and capture. So here, I mean, I don't know. There must be many other molecules which are not part of the spindle. They are not going to be part of spindle, but they must be obstructing the search and capture, right?
Yeah.
So
Yeah.
in spite of the fact that they may be there, is it possible that they are not obstructing, they are not delaying the whole thing? Or, I mean, what is your opinion in this?
Yeah, that's this is a really excellent question also. Of course, chromosome arms are the biggest obstructionist, very likely, because they completely crowd the space. But it's very possible that the following is true. There was actually work in oocyte cells showing that there's like if you take all the actin and myosin, which is on the cell, and generate contraction at the same time with mitosis. So the obvious result is that actin myosin mesh starts to contract centripetally. Mm-hmm. And it normally starts compressing around the spindle, and the result is it actually brings the chromosomes closer to the spindle. So we largely favor this model, and this is actually one of the few alternative hypotheses we are investigating right now. So we much of our quantitative data points out that it would be consistent with the initial stage being this actin myosin mesh compresses chromosomes to the spindle. So we are experimentally investigating it now.
Yeah. Thank you.
I have a question.
Oh.
Can I ask a question?
Yes, sure.
In the model, does ploidy matter? Because these cancerous cells, they divide happily, and they are known to have more chromosomes than, let us say, 23 pairs and so on and so forth. So would that affect in any way, or does it matter?
Yeah, this is another great question. I mean, biggest interesting difference of cancer cells, not only this ploidy problem, but the fact that there's very often more than one centrosome. And so we actually recently had a paper in Biophysical Journal with Raja Paul group, and Apurba, who was asking question earlier, is one of the authors. So there are lots of additional processes happening in cancer cells to deal with those problems, and unfortunately they divide pretty normally even in the face of these problems. So different number of chromosomes, probably not such a big challenge. I mean, who cares if there's 40 or 50, it's kind of the same order of magnitude. But more than two poles, that's of course a big problem, but even that is dealt with. So there's lots of course, processes that going on in cells other than what I told about. Yeah.
Yeah, thank you. Are there any more questions? Please just raise your hand or start to talk. Okay, I don't see any more. Also, the chat is free. So then I would say, well, thank you very much for this fascinating insight into the mitosis machinery. I'm really shocked how this the old model of search and capture sort of is overthrown by experimental data and this new picture. So thank you very much. So I would say you're welcome. Debashish, is there anything else to announce?
I'd like to just thank Alex and Heiko, both of you. So thank you, and we'll meet tomorrow morning Indian time.